So our final speaker of the morning session is Dr. Andrew Mehle, who is a postdoc in Jennifer Doudna's lab and getting ready to start his own faculty position. He is going to be talking about jumping species, how the influenza virus polymerase regulates tropism.
I want to thank the organizers for the chance to come and tell you our story on influenza virus. I also want to acknowledge their exquisite taste in electron micrographs in choosing a beautiful picture of influenza to grace their posters. I also want to give a chance to thank SEND as just being a source of nucleation for this type of work here at Berkeley and across campuses. So today I'll tell you our work understanding and trying to get a handle on how the influenza virus polymerase regulates species tropism. Now, as this audience is well aware, in 2009 there was a new influenza virus that emerged in the human population, and it started in Mexico and was rapidly transmitted in the United States, and within just a few short weeks was global. This ultimately became a pandemic virus. It's the 2009 H1N1 virus, and this movie only shows about 4 weeks. Now, that pandemic occurred and continued through 2009 and through the entire 2010 season, and it was finally declared over in the fall of 2010. This virus infected between 40 and 90 million people worldwide, but thankfully it had a relatively low pathogenicity, causing only between 9 and 18,000 deaths. And this is on par with, or maybe even a little bit less, than your garden variety seasonal influenza virus. At the opposite extreme, however, are bird flu viruses. And these are the viruses you might have heard about in the late 1990s. These emerged in 1997. And while there have only been 500 cases that have since been confirmed, these are viruses you don't want to get. They have around 60% mortality rate, and the death is actually very unpleasant. So what keeps influenza biologists awake at night and the epidemiologists awake at night is that what would happen if you had a virus with the transmissibility of the 2009 pandemic, and even just a small amount of the pathogenicity that's associated with these bird flu isolates. That's exactly what happened in 1918. So this Spanish flu virus emerged in 1918, and it's predicted that it killed upwards of 50 million people worldwide. And controlling all of these events are the ability of new viruses to enter into the human population. And while these are relatively extreme examples, it's easy to forget that each year we have a new strain of influenza virus that comes into the population. And that strain in the United States alone is responsible for between 10 and 30,000 deaths and a huge economic burden and public health concern. So how does influenza continually populate the human population? And to really get at that, you have to understand the virus exists in a natural reservoir, and for the case of influenza, that natural reservoir are migratory water birds. These viruses generally are transmitted as some sort of intermediary species, and in this case, pigs are a prime example, and pigs are thought to serve as a mixing vessel because they can be infected by viruses both from birds and from people. Then these viruses reassort and undergo evolution in pigs, and then ultimately pop out into the human population. And as you go from each of these steps, the virus adapts to its new host and becomes attenuated. Now, as I had said earlier, in very rare cases, you can have transmission directly from birds into people. And these are often pathogenic viruses, so the 1997 bird flu and the 1918 isolates. Now, thankfully, these viruses, especially the more recent H5N1 isolates, are self-limiting. They don't transmit from person to person, whereas these tend to be the ones that become the new circulating strains. Now, all of the viral gene products play a role in this process. I'm going to focus my talk today just on the role of the influenza polymerase. So let me remind you briefly that influenza is a negative sense RNA virus, meaning that it has to first transcribe its genome before it can make proteins. There are 8 different genomic fragments that are packaged into the virion as ribonucleoprotein particles, so they contain a copy of the polymerase attached to these things in addition to the viral nucleoprotein. It's an envelope virus, and upon attachment and entry, it deposits these ribonucleoprotein particles into the cytoplasm. Now, unusual for an RNA virus, it then transits these RNPs into the nucleus. And it's in the nucleus where all the transcription and replication take place. The new viral transcripts program synthesis of viral messages. The genome's also replicated into new ribonucleoprotein particles that are transported to the cytoplasm where you have assembly, budding, releasing new virions, and this process then repeats. Core to all of this, then, is the viral polymerase. And as alluded to in the previous talk, it is a jack of all trades. So for influenza, it is a heterotrimer composed of 3 different subunits, PB1, PB2, PA. It is a large complex of about 250 kilodaltons. The PB1 is the structural and the catalytic core. So it binds to the viral genomes in a sequence and structure-specific fashion, and it also has the actual catalytic activity of the polymerase. PB2 and PA have best defined roles in viral transcription. PB2 binds to the 7-methyl-G cap that's found on host mRNAs. And then PA, as you just heard, is the subunit that undergoes this endonucleolytic cleavage to produce those caps necessary for transcription. In addition to the polymerase, there's the essential viral cofactor nucleoprotein. It's a 56 kilodalton protein that oligomerizes onto the viral RNAs and serves as both a chaperone and a protectant and helps recruit in the viral polymerase during the process of both transcription and replication. And I'll briefly just go through the activities of the polymerase. So the polymerase assembles with a viral RNA, and then it undergoes replication, which is a primer-independent fashion. So I don't show nucleoprotein, but it's also important for this process. So you have a primer-independent replication into this plus-sense copy RNA intermediate that then serves as a template for the synthesis of new viral RNAs in their minus sense. A decision is also made here. Instead of replicating, you can transcribe. And this transcription, as you heard, is a cap-snatching event, where PB2 binds to host messages via their 7-methyl-G cap. The PA subunit then cleaves these caps off the messages, yielding a 9 to 17 nucleotide capped oligo. That capped oligo is then repositioned into the active site of the PB1 subunit, where it primes synthesis of viral messages. As you reach the end of the template, the polymerase encounters a poly-U tract where it stutters and produces a poly-A tail. So in this unique fashion, you have both capped and polyadenylated messages that are priming and directing the synthesis of viral protein expression. Now, not only is this the key regulator of all of the gene expression and genome replication, but it's also a major determinant of species tropism. Where do viruses replicate? How efficiently do they replicate? And is that replication pathogenic? In particular, it's the PB2 subunit that seems to be the main driver in species tropism, and it's just one single amino acid change in that subunit. It's amino acid 627, and that prior to 2009, every circulating human strain had a lysine at position 627, whereas most avian strains contain a glutamate at that position. The structure of this domain was solved by John Cusack's group, and what it shows us is pretty much nothing. There's no difference between these two domains. They're completely superimposable. The only thing that changes is the side chain at position 627. Now, this 627 domain lies outside of all of the known functional domains of the polymerase, and its real function is still an open question and something that we're actively investigating. But what we do know from looking at sequences that occur in nature and then experiments that have been done in the lab is that the presence of this human lysine at position 627 enhances infectivity in model systems. It's associated with pathogenicity in mammalian systems, and in certain cases is associated with a higher pathogenic virus in naturally occurring human infections. What 627 is doing, however, is unclear, and we set out to understand the role of this single amino acid change and how it's regulating species tropism. And to do that, I adapted a polymerase activity assay. And these assays are fairly straightforward, where we can take a viral gene that encodes our favorite reporter, in this case luciferase. In the presence of an active polymerase nucleoprotein, that gene can be replicated, it can also be transcribed, and then we can detect luciferase as an endpoint. We first did this assay in human cells, where we were able to recapitulate the phenotype observed with viruses. That when we contain a wild-type polymerase that has a lysine, we have a high degree of activity, but when we mutate that lysine to the avian glutamate, we now see severely impaired polymerase activity. And it's not just whether it's a lysine or a glutamate. We can expand this a little bit further with conservative changes and go from lysine to arginine and see that those changes are nearly identical in activity, whereas if we mutate from glutamate to aspartate, it's even more impaired. So something about having a basic residue at 627 seems to be a key regulator in activity. The intermediate changes to histidine or alanine show us an intermediate phenotype as well. And this isn't true just for human 293 T cells. It occurs in COS7 cells. And actually, it's occurred in every primate cell that I can get my hands on. You always need to have a basic residue at position 627. Repeating these assays in avian cells, we see a completely different result now. The wild type that has the lysine still functions at a high level, but now this glutamate that was impaired in human cells is highly active in avian cells. And that's consistent with being the dominant residue in the avian population. This really is the avian preferred amino acid at this position. So it's not just that having a glutamate at 627 globally impairs polymerase function, but rather it is a cell type, and in this case, a species-specific defect in polymerase activity. And in work that I won't be able to show you today, we've gone on further to investigate this at a molecular level. We know that all of these proteins are being expressed and expressed at similar levels, whether they're active or not. All of these proteins assemble into the trimer polymerase that I showed you before, and they're all localized into the nucleus. The defect is that avian polymerases can't go from a trimeric polymerase into this higher order ribonucleoprotein structure that's essential for replication and transcription. So that left us with at least two possibilities we could consider. Is it the case that in avian cells, there's some sort of factor that compensates for having the avian glutamate at 627? Or conversely, could it be that human cells have a factor that recognizes and targets polymerases that have the acidic residue at 627, and that you need to evolve around that to function efficiently? To do this, we can't really cross human and chickens, but the next best thing we can do is a heterokaryon, where we can cross together human and chicken cells. So in these assays, you express the polymerase and nucleoprotein in one cell type and fuse that to another cell type that has our reporter. Upon fusion, you reconstitute polymerase activity only if the polymerase is functional. And just as a proof of concept down here, we can express GFP in human cells, RFP in chicken cells, and then see dual fluorescent cell types upon heterokaryon formation. And the first question I asked is there a compensatory factor in chicken cells? And to do this, I expressed the polymerase in human cells. Now, the human polymerase with the lysine is active in these cell types alone, whereas the avian polymerase with the glutamate is inactive. If there was a compensatory factor, we would expect that upon fusion, we would reconstitute polymerase activity for the avian-style polymerase. But in fact, that's not what we saw. We saw upon fusion, the lysine-containing polymerase was highly active, whereas the avian polymerase was still impaired. So there wasn't some compensatory factor present in a chicken cell. So then you obviously can guess what's the next experiment. We did the converse. We now express these polymerases in avian cells where both the lysine and the glutamate version were active and fused them to human cells. Upon fusion, once again, we got high activities for the wild-type lysine-containing polymerase. But now we've impaired the activity of the glutamate-containing polymerase, and it's on the level of background in the absence of PP2 altogether. So recall before that this polymerase was at least 75% active in avian cells, but now when we supply the cytoplasm from a human cell type, we completely abolish its function. So all of these data combined allow us to put together a model where we think about how do viruses move into humans, and what are the factors that the polymerase has to adapt to and encounter as it's moving into a new species. And our model consists of the idea that for some reason that we don't yet understand, polymerases that have an avian-style mutation at position 627 in this PB2 subunit fail to assemble into ribonucleoprotein particles. That blocks virus replication, that lowers pathogenicity, it impairs transmission from host to host. And in our case, you don't assemble into these ribonucleoprotein particles, whereas that exact same polymerase with only a single amino acid change is fully functional in a chicken cell type. So given the importance of this PB2 change that I've told you, one would assume, and we certainly did, that the 2009 virus is going to have a human PB2. But what became very apparent is that these 2009 viruses have a complex evolutionary lineage. They're so-called triple reassortment viruses, and they have gene subunits that were derived from 3 different sources, from pigs, from humans, and from birds. And surprisingly, both the PB2 and the PA subunit have an avian origin, and the PB1 now stays human. So how is a virus that has a PB2 subunit from a bird, which all of our experiments show should be restricted, replicating in humans. Maybe it has a point mutation in amino acid 627. Well, that's not the case. All of the 2009 viruses retain this restrictive glutamate at 627. So the first question I wanted to know was, are these polymerases even restricted? And now I'm using some primary isolates in the lab. And notice that our activity assays are on a log scale here. So we're using polymerases derived from the 1918 virus, one derived from a 2003 isolate from upstate New York, and then an avian polymerase from Ohio in the early 80s. When these polymerases contain a lysine at position 627, they're highly active, whereas when they contain the glutamate, they're impaired by at least 2 and approaching 3 logs. And we can conversely do this with the avian polymerase. It's normally got a lysine, it normally has a glutamate at 627. It's poorly active, mutating it to lysine rescues its activity over 100-fold. Testing 2009 viruses, I cloned 3 different polymerases from 2 isolates from Utah and then 1 isolate from California. Looking at these polymerases, when they have a glutamate mutating it to lysine, we do see that they rescue polymerase activity. So these polymerases are partially restricted, but the effects are relatively minor. Instead of being 100 to almost 1,000-fold, it's as little as a 4-fold effect. So are there other changes in the 2009 virus that could account for these highly active polymerases, even though they contain the restrictive glutamate? And it didn't take long to find. Looking at the sequences, we see immediately upstream of this glutamate at 627, a highly unusual polymorphism that's present in these 2009 isolates. Prior to 2009, all of the humans had a glycine or a glutamate at this position, but now all of the 2009 viruses have this serine-arginine, or what I call an SR polymorphism. Where did the SR polymorphism come from? As I told you, this is a triple reassortment with genes from birds, pigs, and humans. The SR polymorphism first arises in pigs in the late 90s when they underwent this reassortment process that was actually the direct ancestor of the 2009 virus that came out. So it first appears in 2009, then it coexists in this population with viruses that have glutamate. It's never found in a virus that has a lysine at 627. And then these are the viruses that emerged in early 2009 in Mexico. Mapping these changes back onto the structure of that domain gave us a little furthermore confidence that this could be an important change. I'm showing here the human and the avian forms of the 627 domain, just really highlighting residues 591 and 627, and we see that they're juxtaposed in 3-dimensional space. Mapping in then the mutations of this 2009 virus, the arginine, we see that the arginine and the glutamate are right next to each other, raising the possibility that maybe they're interacting, and that could affect the function of these polymerases. And we could return to the polymerase activity to test that directly. 2009 viruses that had their SR polymorphism mutated to the glycine glutamine that was present in humans before 2009 show a reduction in polymerase activity, suggesting that it's an important player in adaptation to humans and escaping from this dominant restriction that we have previously identified in our heterokaryon assays. It's probably not the only thing that's playing a role, and we'll get back to that a little bit later. So if 2009 viruses have acquired this SR polymorphism, can we move that now into heterologous system to see, is that sufficient for adaptation, or are there other changes? We could use a primary avian isolate that we had in the lab, S009, and now introduce the SR polymorphism. This virus is significantly impaired in human cells. When we introduce the SR polymorphism, we see a 40-fold increase in polymerase activity. Introducing a lysine into that virus causes an 800-fold increase. And then having both the lysine and the SR polymorphism doesn't cause anything additive, suggesting that maybe they're functioning on the same pathway or in the same mechanism. So these are in human cells. When we do the exact same experiment now in chicken cells, we see that all of these polymerases function at a similar level. And this is consistent with the idea that the SR polymorphism is escaping the restriction event that occurs as the virus moves from birds to people. From this, we could then suggest a mechanism by which the SR polymorphism is actually functioning. So what our initial data showed was that you really needed to have a basic residue at position 627. Whether it was an arginine or a lysine didn't matter as long as it was basic. When you have an acidic residue, though, it disrupts an otherwise basic face. And this is what we see if there's a lysine at 627. If you introduce the SR polymorphism, it's predicted to partially neutralize that charge and restore, to a large extent, this basic nature at that position. So we predicted that this would be the case. And Yoshi Kawaoka's group has since gone on to solve the structure of this domain with their collaborators and show that it is in fact part of what's going on. As they solved this structure, they did see that the arginine is positioned right next to the glutamate, and it partially neutralizes that charge that's present there. But in addition, it causes a slight conformational change, that in the absence of the SR polymorphism, there's a little bit of a channel or a cavity, but what's happening is the arginine is sitting on top of the glutamate, so they're not going head to head, they're kind of lying on top of each other. And they since have gone on to show that this is important for virus replication. We could look at this a little bit more to try to understand what are the structural constraints around this position. And one of the things that's always been very interesting to me is not only is this glutamate or lysine at 627 very important, there's this highly unusual polyproline tract just upstream. It's proline, phenylalanine, alanine, alanine, proline, proline, or PFAP. And looking at that in the context of the structure, this is again the 627 domain, and we can peel back a little bit and see that these prolines are actually within an alpha helix. And maybe it's positioning this loop where 627 is present to see for function. And using the polymerase activity, we could explore that quite extensively. And sure enough, the initial proline and phenylalanine that are terminating this helix are very important for polymerase activity. Mutating them to alanine impairs function more so than just having the glutamate there. So it would suggest to us that it's both the charge on the 627 domain and the positioning of that residue that's critical for function. Now, none of this matters unless it's important for the virus itself. We could take these changes and put them back into viruses where we could then investigate the function of the SR polymorphism. Viruses that are avian and have a glutamate replicate, but they replicate to significantly lower levels in human cells than those viruses that have either the SR polymorphism or the SR polymorphism plus a lysine. When we do these experiments in pig cells, which are the intermediate mixing vessel, we again see an intermediate phenotype where having the SR polymorphism causes only about a 4 or 5-fold difference in whether or not you replicate. And then all of these viruses replicate to similar levels in chicken cells, consistent with the fact that chicken cells are permissive for any changes at position 627. So in addition to all of these assays that we've done, others have now gone on to show that the SR polymorphism is important for replication in animal systems and pathogenicity in that context. And thankfully, you don't get additional adaptation by acquiring a lysine at position 627. This is consistent with our polymerase activity assays, and now 4 separate groups have gone on to show that those viruses don't replicate any better and don't cause any more disease. In addition to having individual point mutations, influenza evolves by other strategies, notably reassortment. So you have dual infection of a cell with 2 different viruses. In addition to getting back these parental viruses, you actually get back reassortant viruses where 1 or more of the genes have been exchanged between these 2 parental types. And this is a very common phenomena for the polymerase itself. So I show here, and this coloring scheme is going to hold throughout the rest of the talk, avian polymerases and human polymerases in yellow and purple. The pandemics of 1957 and 1968 were polymerase reassortments where the avian PB1 subunit was substituted into an otherwise human polymerase. And then the 2009, as I had mentioned, was actually a complex reassortment where we have avian PB2, PA, swine NP, and human PB1. So could we predict different ways in which viruses might adapt into humans? And would those be similar or different from the mutations that have to occur at glutamate position 627? And using our polymerase activity, it was rather trivial to swap in and out different subunits. And these are data that you've seen before, that in human cells, the avian polymerase is impaired. The SR polymorphism causes a 40-fold increase. And then having both a lysine or a lysine and an SR causes high levels of activity. Reassortments of the human polymerase had moderate effect on activity. So when we introduce a human nucleoprotein, there's a moderate change. And introducing a human PB1 subunit showed a severe incompatibility between these different polymerase subunits. To our surprise, acquisition of a human PA subunit significantly rescued the activity of an otherwise impaired polymerase. So both those with the glutamate and those with only the SR polymorphism are now significantly more active. And this wasn't just the lab strain that we had been using. This was true also for several primary isolates, 1918, NY312. It was also true for the 3 different isolates from 2009 that we looked at. And in fact, the 2009 isolate California 07, that PA subunit was just as active, if not more active, than if we had made the single point mutant from glutamate to lysine. So now there's some sort of second site suppressing effect that's going on between the PA and the PB2 subunit. We could map that first with chimeras, and for time I'll just tell you that the activity resides in the C terminus of these chimeras. So any PA subunit that has a human-derived C terminus is highly active. And we could then map that even further down to a single change in PA. So shown here are the avian residues mutated to their human form. So if I take a wholly avian polymerase that should be restricted in a human cell type and now give it a threonine to 552 serine change in the PA subunit, completely removed from glutamate 627 in PB2, we rescue activity. The structure of that domain has been solved. Unfortunately, again, it doesn't tell us much other than that threonine 552 is going to be surface exposed. So maybe there's an interaction between PB2 and PA, but we don't quite yet understand what that is. So if PA is genetically separable, that we can move it back and forth and reassort, and now we can actually show that these domains are also separable, could we make the protein physically separable? So we tried that. We expressed just domains of PA, the C terminus and the N terminus, and sure enough, we could reconstitute polymerase activity with this bipartite protein. So whether this is relevant to an actual infection, which would suggest maybe you can have transcomplementation, or whether this is just sort of a biological parlor trick is not yet known. But it does suggest the idea that not only could we have complementation and reassortment within a virus, but we could actually get different PA subunits helping each other out. And once more, just briefly, this is important for virus replication. Viruses that acquired a human PA in the context of an otherwise avian polymerase replicate faster in human cells, and those viruses are more pathogenic in that they cause more rapid weight loss in mice, and mice reach their endpoint faster. All of the mice that had a PA subunit died, whereas those that had the wholly avian polymerase showed a delayed kinetics in weight loss, and in fact, one of those mice was lucky and survived. So let me summarize then that what I've shown you is that the polymerase utilizes many different strategies, sampling whether it's its flat evolutionary space or its entire Swiss army, to figure out how is it going to cross species barriers. The major species barrier that we've identified resides in whether or not the virus can address the basic charge on the surface of the 627 domain. The SR polymorphism was a novel strategy used by these 2009 viruses, and we predict that reassortment can also be a strategy in which this virus can, one, replicate efficiently in human cells and possibly make a more potent virus. So this is just summarized here then, where we see all the different ways in which viruses adapt as they move. And let me just finish by acknowledging the fact that all of this work was done here in Stanley Hall in Jennifer Doudna's lab up on the 7th floor with the tremendous support of her and her lab, and then our collaborators Viv Delgan and Jeff Taubenberger, who did all of our animal work. And with that, I'd be happy to take any questions.
So I think the avian hemagglutinin, which is key to binding to sialic acid in the bird, I know we didn't talk about that, but do you think that has a stretch of lysines in the avian form, right, of the hemagglutinin? Do you think that charge has anything to do with the polymerase? You know, that there's the lysines are in the humans but not in the birds?
Yeah, so the question alludes to the fact that as the virus moves from birds to humans, it's not just the polymerase, although I'd like to think that there are many other viral factors that are playing a role. In hemagglutinin, the viral envelope basically is a determinant because the receptor differs from birds to people. There's no known interaction between the hemagglutinin and the polymerase. The functional end of the hemagglutinin is going to be external to the virion, and the polymerase is inside. And the hemagglutinin is never thought to actually get into the nucleus anyway. The polybasic tract that you're referencing, I think, is actually the cleavage site. So hemagglutinin needs to be processed from hemagglutinin 0 to hemagglutinin 1 and 2. And if you can acquire this polylysine tract, then processing can be faster and can occur in more cell types, and that can increase your pathogenicity. So that's a determinant as to whether you can get across species, and if you get across, are you pathogenic? But I think it's independent of the polymerase.
So if I understood your data correctly, the lysine and the SR polymorphisms that enable the polymerase to escape restriction in human cells, they don't seem to have any readily apparent fitness cost in the avian cellular environment, at least in the data that you presented. So the questions I'd have from that would be, number one, is there some selective trade-off, within the avian host, or are you actually seeing, since there's continuous replenishment of the avian reservoir from human and pig, do you actually see that there's an enrichment of those polymorphisms in that host population? And related to that, are any of these mutations in immunogenic domains like CTL epitopes or anything that might kind of present this additional tier of selection that balances those frequencies of the polymorphisms?
Yeah, so why does the avian population have a glutamate? If you look at sequences prior to 2009, roughly about 70% of the bird isolates had a glutamate at position 627, and then there was a bit of diversity. It was relatively rare to actually have a lysine, although there's at least one paper where bar-headed geese in Qinghai Lake in China had acquired a lysine, although that rapidly died out. So I would suspect it's not totally neutral, that there's some reason that the glutamate is sustained in this population. We don't really know what that is. There are many factors that can contribute to it. One major thing to consider is that when we get infected, it's an upper respiratory tract infection. When birds get infected, it's in their gut. So then, alluding to your second question, is there an immunological drive for this? I don't know the answer. If any of these regions are particular CTL epitopes, at least in our case, when we do this in culture, there's clearly no pressure there, and we see the effects that we see.
Last question.
So Andy, very nice data, but I have a sort of a technical question that may be important. In your luciferase assays,
yes,
normally you normalize everything to 100% in human cells or in avian cells.
Yes.
But then the activity in avian cells and in humans could be very different. It could be 100 times different, or 10. In fact, the titles of the virus in avian cells and in human cells are very different. So the question is it really these mutations are tolerated in avian cells, or that in avian cells the virus just replicates slowly, and this mutation doesn't really matter so much, and for other reasons is selected? That's still open.
Yeah, in terms of the polymerase activity assay, you're right. It's very difficult to compare absolute numbers because there are a number of factors that get into it. What we do see, in terms of raw numbers, we're getting hundreds of millions of luciferase counts from all of our assays. So it's very robust polymerase activity. If anything, I would suspect that the viruses are replicating faster in avian settings. The temperature's elevated up to 41 degrees versus 33 in humans. I don't know raw numbers on that, though. And it certainly would have an effect because I suspect, anyway, that the polymerase is going to be one of the limiting components in the virus replication cycle. So if you have a more or less active, faster or slower, it would definitely affect all of the processes of the life cycle.
Yeah, I guess the point is maybe the limiting factor is not the rate of elongation of the polymerase in one system versus the other, and so that's maybe why this mutation is better tolerated as well.
Yeah, it could be.
In addition to all the compensatory mutations that you
Yeah.
nicely show.
Yeah, thanks.